Okay, looks like we're all set to go. So thanks everybody for joining us for the second seminar from our speaker this week, Dr. Pete Marra. For those of you that didn't tune in to his talk yesterday, Dr. Marra is the director of the Georgetown Environment Initiative. He's also the Laudato Si professor of biology and the environment at the McCourt School of Public Policy there at Georgetown. Before going to Georgetown, he was a research scientist at the Smithsonian Institution for 20 years, most recently as the director of the Smithsonian Migratory Bird Center. We're going to hear today about Pete's research on migratory birds, which has been extensive and extremely influential in the field of ornithology over the time that he's been doing research. He's published over 200 papers at this point, many of which have focused on migratory birds and how we can better understand their ecology and evolution by studying them, both on their breeding grounds as well as during their non-breeding season. And I'm sure he'll tell us today that's an area of the ecology of these species that was really underrepresented in the ornithological research for many years. And I think it's not an exaggeration to say people are paying a lot more attention to that now because of Pete's leadership and vision on that topic. So with that, I'll turn it over to Pete, and it's my pleasure to welcome him here this afternoon.
Thank you very much, Clark. Really appreciate that, and really had an enjoyable time virtually talking to many of your students over the last couple of days and learning about all the cool work that's being done at Utah State on a whole bunch of different things, things that I can truly relate to. So it was really a pleasure. So today I want to talk about stuff that really is near and dear to my heart. It's about full annual cycle ecology and how periods of the annual cycle interact. It's something that I've been really working on for the last 30 years in primarily one system that I'll talk about tonight, but I've actually extended this into multiple systems, and not just in birds, but also into herps a bit, and I've dabbled in a variety of areas. Before I do that, one thing I want to emphasize, and I can't really show you a slide with everyone's names because there's just too many of them, but all my work, and I think it's hard for any of us to say that none of our work is collaborative, but 75 to 100 people at a minimum have contributed in significant ways to everything I'm going to talk about. Everyone from the various mentors that have been really influential in my work to remarkable students like Clark in this effort. It really has been an incredible brain trust of people thinking about the problems and ways to deal with them, to the production of the papers, to how we communicate this information to a broad audience, not just to the scientific audience. And we do take that really seriously. To the funders, and my work has been supported by a whole suite of different federal agencies, from National Science Foundation to a little bit of NIH, to the DoD, to corporate sponsors, to individual foundations, and to individual philanthropy. So it's really been a broad effort to get this done. And as you all know, it really requires that. It really requires you to be entrepreneurial to fund your work. You can't count on any one source to do anything. I was part of a team of folks in that collaborative sense, a large team of folks that published a paper last October, actually it was October 2019, almost 2 years ago now, a year and a half ago, reporting major declines in migratory birds, in North American migratory birds, from 1970 to 2017. We reported not just in the number of species that are declining, which was quite significant, but we recorded a net population loss of about 2.93 billion birds since 1970, about a third of North America's birds. And we used multiple different ways to get at this, so we feel like our results were fairly robust, both terrestrial sorts of counts as well as NexRad radar, which independently demonstrated that there was this real significant loss in bird abundance, which has all kinds of implications that I won't go into. But it was around 303 to 529 species, 57% of North America's avifauna was in decline, not just migratory species, but all of the species that are considered North American species. The challenge is that we don't really know where, when, or what is causing the declines for most of these species. Yeah, we know the broad threats. We know that it's probably habitat loss as the big driver. We know that it could be neonicotinoids. It could be overharvesting for some species. It could be cats for other species. But pinpointing where and when that's happening for species that occupy, in some cases, from pole to pole in the Western Hemisphere. Some cases they go to Asia. In some cases they go to Africa. The breadth of those areas covered is just tremendous. And so if you want to try to figure out what's causing these declines, it becomes incredibly complex because it's probably multiple and interacting things that are driving these declines. So implementation we have to figure out where it's happening so we can get a big bang for our buck. And a lot of my research over my entire career, coming out of even my graduate work, has really focused on this fundamental issue. What are the factors that limit and regulate populations of animals? It seems like it's such a fundamental question, which is what I love about it. But I also love that it has such true application to the conservation problems we're dealing with today. So it's that fundamental and applied that gets to the very basic things that I really like to do, that really keep me up at night. So this afternoon, what I'm going to talk about are cycles, this fundamental thing that I'll get into, and how something about migratory connectivity. It's this new thing that I'll talk about, and how it's fundamental for getting at understanding population dynamics and population regulation. I'll illustrate this through an example with one species, the full annual cycle ecology of the American redstart. It's this one species that I've really focused on for the last 30 years, although not the only species. And then I'll take a look at some new tracking approaches. I'll introduce you to some of the other tracking approaches that I've used over the years, but I'll introduce you to some new ones that we are now starting to use. But let's start off thinking about cycles. I love thinking about it from this perspective because when you think about cycles, they are pervasive throughout all physical and biological systems, and they really drive almost everything we see. And there's many different types of cycles, but the one I really kind of want to focus on is the biggest of all that I think is the biggest of all, and that is the rotation of a tilted Earth on its own axis, but also around the sun, that drives not just these light-dark cycles that we are slaves to, and so are many animals, but it's also the annual cycles of our seasons. And we watch and we live through a winter that brings us into spring, that brings us into summer, and then fall, and back into winter. But it's these cycles that consequently is a driver on the behavior and the physiology of almost every living thing on the planet. And it's not just always seasons of temperature, it can be seasons of precipitation around the tropics. But it's really critical in driving these circadian and these circannual cycles at the biochemical, at the cellular, at the organismal, at the population level. It's really fundamental. It's what drives, in many cases, migration. Now, I'm not going to argue that we haven't paid attention to cycles in some areas. We certainly have in the biochemical areas, in biochemical fields, in the geological fields, in endocrine fields. But I am going to argue that in migratory animals and in ecology, in many parts of ecology and in evolution and in conservation, it's only been recent that we started to pay attention to the role of cycles in driving important biological phenomena. So I decided to look at this, and I, along with several postdocs, quantified, we did a review, and we quantified when in the annual cycle research was done on 4 different vertebrate taxa: amphibians, reptiles, birds, and mammals. And we just basically did a lit review, looked at the literature and to find out what season was most research done, how many of those studies actually looked at their animals in multiple seasons, and how many considered something I'm calling a seasonal interaction. That is, when they followed the animal or followed some facet of the animal's biology from one season to the next to try to understand how the variance in one season was explained by things prior to that season, in other words. And what we found was there was, in fact, and not surprisingly, but this quantified it, there were severe biases and across all vertebrate taxa. It varied for some, less so in mammals than in birds and amphibians and reptiles. But in general, most studies were done during the breeding season. Most studies focused on one period of the annual cycle, and very few studies considered seasonal interactions in the biological phenomenon or the variance that they were trying to explain. Why is there a bias? Well, I think there's a bias for several reasons. The first of which, it's just logistically challenging to study animals throughout their annual cycle. It's expensive, and the timing is difficult because, let's face it, folks, most research is done by our students, by postdocs, during their academic semesters in temperate areas, which is where most research has been done. They're in school. They're in school in the fall and the spring, and it's focused. Most research is focused during those summer months, and it automatically biases us towards those breeding season components. It's just a hypothesis. I didn't really test that, but I think that's probably true. A second reason, and some pretty famous biologists have said this, ecologists, there's this false impression that everything outside the breeding area is unimportant. It's all about reproduction. It's all about sexual selection, those sorts of statements. They're in the literature. But there's also a third reason, and that is that we have a poor understanding of this thing called migratory connectivity. Basically, we don't have good tracking information for most animals because they move. And when they move, we can't follow them. So we've been studying animals for the most part. I agree. You're carnivore biologists. I know there's a lot of you out there. You guys have been ahead of us and ungulate biologists. You've been able to study your animals more recently or for a longer period of time with tracking devices. But for at least the smaller bodied animals, it's a trickier thing. We try to take a little snapshot or grab a little window of information from an animal for a very short time and try to scale that up for their entire biology. And it's a hard thing to do. This phenomenon called migratory connectivity is something called it's the geographic and temporal linking of individuals or populations between different stages of the annual cycle. And this includes breeding, migration, and winter. And it doesn't necessarily have to include a migration. It can be just an animal that's resident, that's moving, but it is understanding how that animal moves across different temporal scales. That's sort of the key thing. There's lots of reasons to study this. It's really critical to understand migratory connectivity, both for fundamental biology and for conservation. And I want to show you a few examples of why it is so important. The first I want to explain to you is about movement ecology. That is tracking animals throughout their annual cycle and what you can learn. This is a story about tracking the migrations of black-bellied plovers. This is Autumn Lynn Harrison. She's a biologist. She manages something I started called the Migratory Connectivity Project. She's on the North Slope of Alaska. You can see there are 3 eggs there. She's got a black-bellied plover. And we attached a 5-gram satellite tag on this bird. This was from a couple of years ago, and she let it go. Let's watch what happens. So this is on the Colville River Delta. Breeding is just finished, and the bird starts to move. Fattening up, young have probably already dispersed. The pair bond is broken. This is just one individual. There are thousands of black-bellied plovers there. It starts to move, makes a big migration jump down to Saskatchewan. Fattening up there for several days, makes one jump, and then makes a big jump. Doesn't even stop on the Texas coast. So most of its fat was probably gained in Canada, and then goes down to its wintering grounds, where it finally arrives in November. It's staying in one general area, might move again, but we can understand things about its speed, how rapidly it's moving, where it's really spending time. We can put remote sensing maps on there. And then it takes off and heads north again, stopping in Matagorda Island. on the Gulf Coast. It's there for quite a while, dodging peregrines, and then makes its jump on spring migration right back up to the Colville River Delta in Alaska for another breeding season. Now remember, that's just one bird. We've actually tracked them throughout their range. And look at the variation of where these birds are wintering. Totally different countries, mainly all coastal habitats, but in very different types of coastal habitats, with very different temperatures, very different climates. This is migratory connectivity. And it's critical to understand, not just for understanding various aspects of its movement, but also for the conservation. I'd like to show you an example of work being done by one of my postdocs, Callie Stanley. That's a yellow-billed cuckoo. She had just attached, this time, a 2-gram satellite transmitter to the bird's back. You can see this is the yellow-billed cuckoo range. This is a breeding bird survey map. You can see all those areas in red. Those are areas that have had severe cuckoo declines. And in fact, in the West, there were Western cuckoos, and they still do exist in really, really low densities, but you can't even see them on here. We would love to capture cuckoos in the West, yellow-billed cuckoos, this summer, but we can't actually get permission to do it in Idaho because the numbers are so low. So we have worked in Southern California and Arizona. Each of these black dots represent areas where we deployed these 2-gram satellite tags. This is a project that Clark is actually also involved in. But we just wanted to figure out where they were going. So pay attention to the dots in the West versus the dots in the East. Look at how the timing is very different. Eastern birds arrive first, then the Western birds come back down. A couple birds go out to Brazil and then come back in. And then the transmitters, for the most part, started to die. These 2-gram satellite transmitters don't last the entire winter, unfortunately. So those were the only tracks we got, but we were pretty happy with where they told us these birds went. What we thought before was that this blue area was the entire winter range of yellow-billed cuckoos. What we just determined was that this is actually the winter range of the yellow-billed cuckoo with our studies. And this is an area known as the Gran Chaco. We then did some analyses on eBird data. I assume most of you know what eBird data are. And it actually told us the same thing without the tracking data, that the wintering population, this is a model eBird range, occurs in almost the same exact area with using citizen science birding data from South America. The problem is that in this area, here's the maps. Here are the western or the eastern birds in the north and the western birds in the south. So they don't even overlap in their range, at least for this small sample size of birds. What's so telling is that in this area, tree cover loss has been really significant in northern Paraguay and northern Argentina. Not so much in southern Bolivia, where the eastern birds appear to be wintering. And most of this cover loss is for soybeans and for cattle grazing. There's very little regulation of land use change there and what happens for farming in these areas. So from a conservation perspective, I hope that is really clear as to why understanding connectivity is so critically important. So it's really important for movement ecology, it's important for conservation, but it's also critical for population dynamics. And I'd like to, this evening, really explain to you why understanding connectivity for linked populations is really critical. And it's basically for this reason that I'm going to argue, that is that periods of the annual cycle are completely inextricably linked, that you can't just study an animal during the breeding season and expect to understand all the drivers of the variation that you see during that breeding season. That's possible, but my guess is it's not probable. So this life cycle diagram illustrates the basic annual cycle for the American redstart, this little beast here in the middle. So American redstarts in, say, the East Coast, in a place like Maryland, arrive on their breeding grounds in around mid-May or early May. And they'll be there for about a month or so, 2 to 3 months breeding. And then they'll go through a molt, a full body molt. They go through one full body molt where they replace all their flight feathers. And then they'll migrate, a fairly quick migration, about a month, down to the wintering grounds. And you can see it's on their wintering grounds where they spend the majority of the annual cycle, up to 8 months, and then migrate back north again to their breeding grounds. And so what I'm going to be arguing with a series of different types of data is that if we want to understand the fundamental biology for an animal that moves like this, and frankly, even one that doesn't move, but if we want to understand the drivers of survival, the drivers of reproduction, natal dispersal, population dynamics, range dynamics, we need to study these species and these phenomena in the context of their full annual cycle. So knowing something like migratory connectivity and where individuals and populations go, so understanding what they're exposed to from a climatic perspective, from any stressor perspective, is essential. It really is essential. I know that I hope it sounds really intuitive, and it's like, well, of course, because it should. It really is critically important. So what I'm talking about are something that we've termed seasonal interactions. These are circumstances within one period or season of the annual cycle that influence events or traits in a subsequent period. And I think of these happening at both ecological and evolutionary scales. At the ecological scale, you can think of them happening to the individual and at the population level. So at the individual level, you can have something we've termed a carryover effect, where an individual might lose mass, body mass, might change in condition, might be better, might be worse, but it might influence when an animal might depart on migration or get back to the breeding grounds early or perhaps late. Or, this concept has been, is also parallels this other concept called capital versus income breeding, which is essentially this same idea as was articulated for primarily for waterfowl and where they get most of their protein and fat stores for egg production. This has been around for years. It can also refer to population density dependent sorts of things at the population level, such that if you have really high reproductive success in one season, you've got a much larger recruitment of individuals down to the non-breeding grounds where you could have negative density dependent effects occurring. And you could think of this as being negative or positive at any time of the season, depending upon the total number of individuals that might be present in a particular place. And that strength of migratory connectivity would also play an important role if individuals are staying in the same place versus spreading out. That could also influence the predictions around density. Seasonal interactions, of course, can also occur at the evolutionary level as well. Think of body size or plumage, or something like a bird that puts on a really bright plumage or elaborate tail feathers or whatever it happens to be in the breeding season for sexual selection. Those might be really counter-selective or maladaptive in another season because of the way they attract a predator. So that animal has to change its look, in other words, so it becomes more cryptic. Those are things that are happening throughout the annual cycle to select for certain traits that are evolutionary in origin. So as we dig into this a little bit more, I wanted to provide you a bit of an ecological backdrop of what we're looking at in this system. And we'll start off by looking at the non-breeding season, the ecology of the non-breeding season first, and what we're going to be testing along the way to illustrate what's going on with birds during the non-breeding season and how does that translate into potential impacts to these birds either during migration or on the breeding season, during the breeding season. And to do this, I will be talking about the interaction between density independent effects as well as density dependent effects that are influencing redstarts. So from a density independent effect, I'll show you how habitat and rainfall can influence food in the form of insects or arthropods, and how arthropods can influence the body condition of birds directly and their ultimate departure and survival. But it can also work through density dependent mechanisms through crowding and work that way. So in the end, I'll just tell you the answer. It is going to be a combination of density independent and density dependent effects that determines the ultimate condition of these birds at the end of the winter season. Redstarts are pretty easy to study in the non-breeding period. They're a pretty common species. They are totally insectivorous. Males and females both hold territories for the most part. There are some birds that move around at larger spatial scales. In the upper part of your, on the right-hand side of your screen, this is an adult male redstart with that black and orange plumage. In the middle, this is a female, an adult female. And this is a young male in the lower right. Young males look like females. And so it's called delayed plumage maturation. I won't go into that tonight at all. So in general, they're easily sexed in age. So for this sort of demographic study, it's quite nice to keep track of sex and age, and you'll see why in a moment. Most of this work has been taking place in Jamaica. Jamaica is in the Greater Antilles. It's just south of Cuba. It's a fun place if you haven't been there. And I've been working in southwestern Jamaica in a place called Font Hill Nature Preserve, although it's hardly a preserve. We've had to fight various powers that be in Jamaica for years to keep it from being plowed over. And it's quite the fantastic habitat. We work in two primary habitats, black mangrove swamp and also in dry scrub. When we arrive in the fall or midwinter, the mangrove is fairly wet. And as that dry tropical season progresses, as you move from January to February to March to April, that March-April period tends to be much drier. Not in all years, as you'll see. But the mangrove still stays fairly moist. It doesn't get quite as dry as the other habitat we've worked in, which is the logwood scrub. And that logwood scrub starts off as being fairly lush, like this, and then it dries out and becomes quite harsh over that tropical dry season. Now, the thing I want to make sure you understand is that redstarts occur in a whole suite of different habitats, not just in the mangrove and the scrub, but from an experimental design perspective, we really focused on these two habitats to work in. What we basically do, we've done lots of things over the 30 years, trust me, but the things that we do in general is we catch birds. We catch them pretty easily, and I can tell you about that a little later if you're interested. We use mist nets. We band them. We measure them in all sorts of ways, their legs, their wings, their bills, their tails, their pectoral muscle, and we weigh them to the nearest 1/10 of a gram. We not only put aluminum bands on their legs, but we also put unique combinations of color bands on their legs so we can watch individuals and map out their territories the old-fashioned way. We used to do it the old-fashioned way. Now everything is pretty much automated for tracking. And then we recapture them multiple times and we weigh them to look at mass change. But we've also I've looked at corticosterone change in the same individuals. I've done telomeres. We've done a variety of plasma metabolite studies. So I'm not going to go into all those things, but if you have questions about that, I'm happy to answer those questions at the end. And then we also recite the birds every three days to determine departure, when they depart on migration. And then we recite them in every subsequent year to estimate apparent annual survival. So we have overwinter survival estimates, and we have apparent annual survival as well. And then we also measure food through a standardized process. Although I will also say that now we're doing fecal barcoding to really estimate and really understand what these birds are eating. And it provides a really amazing picture of what we didn't know before and what we really know now. It's really pretty impressive. Complicated, but impressive. Here we go. For some reason it's not switching. Give me a second. There we go. Too fast. Hold on. So I'll just basically tell you the answer, what we found over the years. In black mangrove habitat, basically performance of redstarts in each of these habitats is totally dependent upon habitat occupancy, regardless of sex and age. So in black mangrove, food availability over that winter period. So we get there early, we go there late, we're there the whole time, but we have these two different sampling periods, sometimes multiple sampling periods. But as you go from that wet to this dry period, in the black mangrove habitat, food for redstarts is high and stable. Whereas in the second growth scrub, it declines over that winter period. Body condition of birds as measured through multiple ways, including just weighing the birds over that winter period, the same individual birds, and also a population sample. And these birds have no obvious subcutaneous fat. So we're looking at protein, but we also look at pectoral shape and mass, and you can actually see how the pectoral muscle just shrinks and shrinks and shrinks. So these birds are burning up protein over that winter period, but it's maintained in the birds in the mangrove habitat, males, females, young and old birds, but it declines in birds in the scrub habitat. So it's clear habitat effect. Same with departure time. Birds in the black mangrove habitat depart early. Birds in the second growth scrub depart late. And annual survival is high, and it's low in the second growth scrub. I should say that overwinter survival is pretty much constant across both habitats. So we're not seeing differential mortality in these birds over the winter period. And you're probably saying, Pete, this is just correlational. Well, it is, you're right, but it's over many years. And it does vary by year, and I'll talk to you about that in just a minute. But we've also done several different experimental things here to look at this experimentally, including with one student, Colin Studs, where we did removal experiments. We basically pulled birds out of the mangrove habitat and moved birds in. They automatically went in because of territorial exclusion. And we actually also traced that using stable isotopes. And I'll explain that in just a moment. But these birds that moved into the mangrove habitat compared to the scrub habitat maintained their mass, had higher annual survival, had earlier spring departure. So clearly a habitat effect demonstrated experimentally. Nate Cooper, another student, actually reduced food availability, insects in the mangrove habitat, and demonstrated the same thing. He actually got these birds to become floaters and vacate their territories by reducing food to what the food levels would be in really dry years. And then we also did an irrigation experiment. A postdoc, Scott Wilson, irrigated plots and compared them to control scrub plots, and basically found that when you add water, you get green leaves, and not surprisingly, and you get insects, but unfortunately was not able to get a bird effect because the area that you actually have to, the amount of water it takes to do this on a territory of a redstart is a tremendous amount of water. We couldn't find that amount of water. So we did get partial effects, but not the bird effect in that experiment. So as I mentioned, it's not just a habitat effect. Different years vary depending upon how much rainfall there might be. El Nino, La Nina years, they all vary on 3 to 5 year cycles. And so we also can look at how performance varies with annually. And on the x-axis, we have March rainfall. The red dots are scrub habitat. Each dot is a year. The blue dots are mangrove habitat sites. Y-axis is arthropod biomass. And you can see in years of, in wet years, arthropod biomass goes up in both habitats, but it always is higher in the mangrove habitat compared to the scrub habitat. But there's a relationship between rainfall and food availability for redstarts. And when you have more rainfall, you actually have birds in better condition. So birds, this is the 0 line. So these are birds that would be right on this line. They weighed the same exact amount in the multiple times that we captured them. And so if you're above this line, that means you're heavier the second capture relative to the first capture. But you can see for both birds in the scrub as well as birds in the mangrove, in wetter years, you are maintaining yourself in a better condition than you are in a drier year.
Hey, Pete, can you hear? We're not hearing you right now. Oops, look like we may have lost Pete. hopefully he'll log back on in just a second. Okay, he just sent a text that he lost connection and he's logging back on. He told me to dance or something, so sorry, I'm not going to do that though. Okay, well, we'll just see if we can get Pete back on in just a second. Hopefully he'll be able to reconnect. Hopefully everybody can stick around for a minute while he figures out what's happening on his end. Well, hopefully he'll get back on in just a second, and we'll see what happened. In the meantime, I guess we can just,
Hello.
oh, there he is.
I don't know what happened. I was expecting you to dance or tell some jokes.
Okay, I just finished the talk for you. I told everybody everything about redstarts that they need to know.
Good. I'm so sorry, folks. I have no idea what happened, but I guess it's that's what happens sometimes. Hasn't happened to me before. All right, let me get back to the main body here. All right, so let's start here. All right, so basically what I've just showed you is that there are a variety of density independent factors. Can everyone hear me okay? Clark, you there?
Yeah, you're good.
Okay, so there are a variety of density independent factors that drive the condition of birds. Habitat, rainfall in different years that influence food, and this influences body condition and departure. Now, what about density dependent sorts of effects? You'd be really surprised at how few people really try to look at density dependent effects, even though it's really fundamental to a lot of what we do. And because this is a long-term study and we keep track of density and a variety of other things, we're actually able to do that. And so we just simply ask that question: does redstart survival, annual survival, decline with density during the non-breeding season? And what we found was that in fact it does. Density declines. You can see on the x-axis is density, apparent annual survival on the y-axis. Mangrove ASY males here and mangrove ASY females here. Both, as density increases, survival goes down. So they exhibit pretty strong negative density dependent feedback on survival. In scrub, it's actually doesn't occur. It's actually weak negative density dependent feedback on survival. And so we decided to sort of look at this and say, well, to what degree does redstart condition decline with density? And does that influence survival later on after they leave? Because we're not seeing differences in overwinter survival with density, but does condition decline with density? And sure enough, we do see, this is birds in mangrove, the densities on the x-axis, corrected mass on the y-axis. So birds up here are in better condition than birds down here. And in mangrove habitat, sure enough, as density increases, the corrected body mass of birds goes down, as we would have predicted with density. But not the case in scrub, which is quite interesting. In scrub habitat, as density goes up, corrected body mass actually goes up as well. And it only barely goes above the zero line in the really high density years, at least high density years for scrub. And our interpretation of this is that in scrub, birds are just trying to settle at the carrying capacity of the environment. they are so limited by food in that habitat that they're just not limited by density. They're more limited by food than they are by density. And these results tend to hold that up. So it looks to us as though a combination of density independent and density dependent processes structure things going on during the non-breeding season in this system of birds. We've learned a variety of other things in the last few years that make us think that this is a little bit different, that birds aren't always as territorial as we thought. Now that we have automated ways of tracking them, I'm happy to tell you a little bit more about that as we in the question and answer period. But what I haven't done yet is I haven't really explained to you how those things happening during the non-breeding season carry over to influence things back up on the breeding grounds. And when I was doing this work in the late 1990s, I guess, in early 1990s, we weren't able to track these birds. But along came stable carbon isotopes and nitrogen isotopes, and it allowed us to start asking these questions. So I asked the question at the time, does non-breeding habitat influence reproductive success back on breeding grounds? And I use carbon isotopes to do this. So if you look at carbon isotopes across a wet to dry moisture gradient, dry being my scrub, the wet being my mangrove, what you find in the plants and then in the insects and then in the redstarts is that insects and birds, bird blood, bird tissues in the dry habitat tends to be enriched in delta C13, so more positive, whereas insects and birds in the wet habitat tend to be more depleted in delta C13 in those wetter habitats, in part because there are different ratios of different ratios of C3 to C4 plants, more grasses in the C4 or in the dry habitats, but also because the water use efficiency of these plants in these different habitats tend to retain or release these larger molecules of carbon. Here's different habitats: wet lowland forest, black mangrove habitat, second growth scrub. This is the delta C13. And again, this is from the blood levels, from blood in the redstarts that we sampled, both in Jamaica, but also I went to Honduras, part because I wanted to go to Honduras, but I also wanted to test this idea in a different habitat and a different country. So we looked at the same relationship there and found the same thing. As you go from wet to dry, you get a more enrichment in your carbon isotopes in the blood and also in the insects in these habitats. And why this is useful is because you can sample birds on arrival, because those birds from the wet habitats and the wintering grounds leave first. We predicted that they would get back to their breeding grounds first, and then we could then track their reproductive success there. And when I first did this back in 1998, I sampled birds as they arrived at the Hubbard Brook Experimental Forest, clipped their toenail, took blood, did a variety of things, and predicted that birds coming back early would have delta C13 values that are more similar to birds from wet habitat compared to birds coming back later that would be more similar to birds in dry habitat. And that's exactly what I found in that 1 year. But I've since found this in many years, and also other students have found this in not only in redstarts in other sites, but also in several other species. So we think it's a fairly robust thing, and we've done this with tracking tools now as well. But what's really neat is that you can then follow the reproductive success of these birds over the breeding season relative to not just their breeding season variables, but relative to their non-breeding season habitats that they occupied, wet to dry. And what several of my students have found is that birds that are in wet habitat tend to produce more offspring. And we've looked at this with extra pair paternity and multiple nests, a whole suite of different things. Those birds that get back early and in better condition produce more young, fledge more young than birds coming back later from the dry habitat. So there's definitely a condition, but more importantly, a timing factor that's driving this reproductive success, which is all started and carrying over from that non-breeding season period. And again, we've demonstrated this using light level geolocators on birds on the breeding grounds. So we know exactly when they've left the breeding grounds and when they've left the non-breeding grounds and when they've arrived back up on their breeding grounds. So we've got the exact time, and we've got isotopes now. And so we've been able to validate this for multiple species, including ovenbirds, curtains, warblers, and now even American redstarts. So let's ask another question that's more of a population level question, and ask whether or not annual survival on the wintering grounds, going back to this wet, dry year thing, differs across these wet and dry years in Jamaica, and does this vary with migration distance? Now, the thing you should know is that the connectivity of our redstarts in Jamaica is actually, it's fairly strong, but birds go to different latitudes. So we've got this fairly wide latitudinal variation in our redstarts in Jamaica. And so here's an isotopic map of deuterium, the isotope of hydrogen. And what's really neat is it varies latitudinally, and you can sample tail feathers that are molted on the breeding grounds of these birds, and they will match fairly closely the deuterium signature that's at that latitude. So if you sample a bird here, it'll be around 70, negative 75, plus or minus a few parts per mil. But what's really neat is that this redstart that might be at this latitude, once it goes down the wintering grounds to a place like Jamaica, I can pull its tail feather, and because it's inert, that isotope is inert, I can determine the isotopic signature and figure out what latitude that bird bred at the previous year. So I can determine its origin the previous year using stable isotopes. Now, I've been doing this for 30 years. I've been collecting these feathers from every bird we sample, hundreds of birds every winter, and we've been saving these for over 30 years, which is pretty cool. So what we decided to do was to look at the origin of birds the previous year, so the breeding latitude they came from on the x-axis, against their apparent survival on the y-axis after wet years and after dry years. I hope that makes sense. And what we found was annual apparent survival was the same across all wet years. But when we modeled this with birds after dry years, and I'm just summarizing these into lumping all the years, after dry years, the latitude you're going to, or the latitude you came from, and they show site fidelity back to those same sites, we know that's the case, varies depending upon where you're going. So birds that are migrating further north have much lower survival after dry years than they do if they were migrating further south in their breeding range. This is pretty important. This suggests that the climate, the precipitation on the wintering grounds can determine what's going on with range limits of these birds back up on breeding grounds. So we can then ask the question, well, because there's been this Caribbean drought that's gone on for 20 years and it's going to be going on for another 20 years, has that influenced the breeding ground origin of redstarts wintering in Jamaica over the time frame that I've been studying these birds in Jamaica? So we basically looked at our deuterium over, I think we did 20 years, to see if the mean origin, the mean breeding latitude of our birds has changed over time because of this drought and because of the impact that it has on annual survival of birds migrating further north. And that's exactly what we found. So going back to 1990, all the way out to 2015, this is the deuterium or natal origin, the latitude that the bird had come from that previous year. This is just SY birds, but we've also done this for ASY birds as well. And you can see that the mean breeding latitude has basically moved south for our non-breeding population in Jamaica. And if we were to map this, basically this is where birds were in 1990. This is where birds are in 2015. This is particularly interesting to me for several reasons. First of all, most climate change models are predicting that in general, birds are going to be moving towards the poles, and a variety of things are going to be moving towards the poles. Well, for most of these predictions, they haven't incorporated events from throughout the full annual cycle. They've totally ignored that birds are actually on the wintering grounds for most of the annual cycle. And what happens there drives fundamental aspects of their biology, including selection on individuals that might be moving further north. When the reality of it is that things that are happening on the wintering grounds appear to be driving various aspects of the range dynamics of redstarts. This is in prep. We haven't published it yet, but we're pretty excited about those results. We're working them up now. So I hope I've convinced you that seasonal interactions drive basic biological processes, not just in redstarts, but we've shown this in various species, and lots of other researchers have shown this in not just birds, and also in mammals and a whole suite of things. We've shown it influences migration schedules, reproductive success, perhaps breeding season range limits, and that full annual cycle perspectives are really essential. We're now trying to figure out most of our work has been about winter influencing migration, spring migration and summer. We're now trying to figure out how summer events might influence fall migration and getting back onto the wintering grounds. It is a full annual cycle perspective. We just haven't figured out how to do that yet with redstarts. And in order to do that, you need really good information on migratory connectivity. So you're working with not the same individuals, at least the same populations. And while these stable isotopes are fun, tracking individuals and using automated telemetry and a variety of things is so much more satisfying. And that's the direction that this work is moving. And we've just in the past few years started to use these light level geolocators, which basically on the backs of redstarts, they weigh about 0.3 of a gram. So redstarts can carry them, and redstarts are pretty tough birds. So we're getting really high return rates, but there's a little light sensor on the back of the bird, and there's a clock, and it keeps track of time and light levels. And you can actually figure out your latitude and longitude plus or minus a couple hundred kilometers. And it turns out our redstart population, we've had the latitude right, but stable isotopes, deuterium, don't tell you anything about longitude. Most of our birds are going towards Wisconsin and Iowa. One bird went to New York, and one in Michigan or so, in Illinois, but most of our birds are going over into this area, which is pretty exciting. The other thing I wanted to tell you about was a totally new technology that we're using called MODIS, which is a nano tag, a coded tag that transmits not to a satellite. It doesn't archive the information, but it transmits to a tower. And we can there's towers coming up all over the place. And we've started to use these with Kirtland's warblers that winter in the Bahamas. This was an endangered species, and it breeds up in the upper lower peninsula of Michigan and in jack pine habitats. There are a few that are in Wisconsin and in the southern part of Wisconsin. But we have now covered one of their 4 islands that they occupy in the Bahamas with antennas and towers, and also their entire breeding range in the upper lower peninsula of Michigan. So we can, and we also have towers along Florida. So we can tag birds here, track the same individuals as they move and hit the coast of Florida, and also detect when they get here. And so we're tracking the same individuals and catching the same individuals in multiple times of the year, which is pretty unprecedented for a small songbird. So we're really starting to learn some pretty cool things about these species. So I'm going to stop there. I'm sorry about that glitch, but I think I've used up my time, and I'd rather spend some time taking some questions. I hope that was enjoyable and clear, but if you have any questions, let me know.
Great. Thanks, Pete, for another good talk. And